Any questions before we start over everything we've already covered? Okay, so we're going to get back into a little bit of talking about cells in particular. And cells come in two types. The first are called prokaryotes. Basically, yes, ma'am.
Just spell
Prokaryotes, prokaryotes. The second type are called eukaryotes. Eukaryotes. Every living thing fits into one of those two categories. Eukaryotes are plants, animals, fungi, and protists. Plants, animals, fungi, and protists. Protists are organisms that just don't really fit with any of the other three main eukaryotic kingdoms.
How do you spell that?
Which one?
The one after fungi. The one after fungi, the fourth one.
Protist, Protist. And the prokaryotes, we are by far most familiar with bacteria. However, there is another group called the archaea. The archaeas are all extremophiles. If we say something is extremophilic, we mean a lover of extremes. These are the things that you'll find living in a hot water spring, for example. Thermophiles, they love heat. They have found them growing, actively metabolizing in ice. Those are cryophiles, C-R-Y-O, cryogenics, comes from the same root, it means cold loving. Halophiles, H-A-L-O, they love salt. Xerophiles, X-E-R-O-P-H-I-L-E, xerophiles. They love the dry conditions, bone dry. Archaeas are thought to probably have been the first living organisms. And when I say extremes, I do mean extremes. Those hot water springs are literally just at boiling temperature. Y'all have heard of the Dead Sea, haven't you? Believe it or not, it's actually not dead. Archaeas. I'm not saying there's a lot of them, but they live there. You get an idea, I think. Xerophiles, you've got the Atacama Desert. It's in Chile and Peru. It's on the west coast of South America. It gets about an inch of rain a century. That's how dry it is. You'll find archaeas there. Bacteria. Bacteria can be found everywhere. The key term there is ubiquitous. There will be bacteria on my tablet, my phone, that rag, desktop, your skin. A lot of people get kind of germophobic about it, but simple fact of the matter is they are living on you, they are living in you. You're actually carrying around probably 1 or 2 pounds of bacteria in your gut. Didn't realize that, did you? The appendix, remember how they've always said there was no function for the appendix. Actually, there is. It serves as a safe haven for the bacteria that live in your gut. And you actually need those bacteria to have a healthy digestive system. In fact, there is a legitimate actual treatment called a fecal transplant. Y'all ever heard of that? It sounds gross, it sounds sophomoric, it sounds juvenile and middle school humor, but it's true. There are certain people who are suffering so bad from things like irritable bowel syndrome that they will actually take poop from a different person and transplant it into the person who is suffering. Because with that transplant come good bacteria. They have actually demonstrated in several cases that's a helpful treatment. Yes, ma'am.
They couldn't just use probiotics? They couldn't just use probiotics or
No,
prebiotics?
because those pass through the digestive system. So you have to take into account the vector as well.
So it's almost like you're transplanting the bacteria.
Yeah, that's exactly what you're doing. But unfortunately, since they essentially live in the colon, it pretty much has to be colon to colon. You know, I'm not trying to gross you out. I just like bringing in the weird stuff because we live in a fun, weird world, don't we? So those are the types of cells. There are two kinds of critters that challenge that. Well, three actually. Number one are viruses. We've already talked about them. You can argue quite convincingly, I think, either way, that viruses are alive or that they are not alive. The main thing to remember about a virus is that it does contain genetic information. It has RNA or DNA, one or the other. It may have to invade a host cell to use its information, but it does have genetic information as we know it. I talked about parasitic reduction, didn't I? Okay. To tell you how weird viruses can get, here's a couple of strange examples for you. There is one known case where a virus infects a virus. Y'all ever heard of this story? There is a fairly large virus called megavirus. It actually has a membrane around it and some molecules. So it's almost like a prototype cell. It's not quite a cell though. It can't survive or reproduce on its own. It will infect a species of amoebas. Then it goes into the lytic cycle, just like we talked about, explodes the amoeba and the virions get distributed. There is another virus called minivirus. Minivirus also infects that same species of amoeba, but it doesn't do anything to the amoeba unless the amoeba already has a megavirus infection. Or if a minivirus infected amoeba then gets subsequently infected by megavirus. In that case, when the amoeba explodes, it's not sending out megavirus virions, it is sending out minivirus virions. How wild is that? Like I said, you can argue either way you want, but everything I see pretty much says to me the viruses are just extremely reduced parasites. That one is so reduced it doesn't even bother having a cellular host. It just takes over another virus. Which brings us to one of the other two things that challenge the very definition of life. These are called transposons. Transposons. Related to that are retrotransposons. Retrotransposons. And as you might imagine, the retrotransposons use RNA rather than DNA. So what is a transposon? It is a naked strand of DNA that moves from one location in the cell to another. So if you have this transposon on chromosome number 1, it can actually cut itself out and paste itself into chromosome 7. In some cases, rather than a cut and paste, it will do a copy and paste. It literally is nothing but a sequence of DNA that moves around the host genome. Almost like it's a virus that's lost the ability to live free. Pretty wild, isn't it? Then we've got the last group of things that challenge the definition of living. And this might be the biggest horror story in biology yet. This is a group of proteins called prions. 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 I see Simon smiling and nodding. You're in the know. It is pretty rough, isn't it? Prions were discovered in the early to mid 80s. Now I want to re-emphasize, they are proteins. They do not have any genetic information of their own. But they act like disease particles. You can actually get a prion infection from another organism that's infected with it. What's worse, it appears they can cross species boundaries. The way a prion works is it comes into contact with a normal protein and causes it to misfold. Causes the normal protein to misfold. The misfolded protein can now act like a prion itself and cause the next one in the chain to misfold. You see how this goes. There is a disease in sheep called scrapie, S-C-R-A-P-I-E. You ever heard of it? Carol has. She's the only one. No reason you would. It causes the sheep to act in bizarre fashion. They would typically scrape their wool coat against a barbed wire fence until they essentially skinned themselves. They also engaged in all kinds of very bizarre behavior. When they did a necropsy on the bodies of these sheep, they found that the brain of the sheep was like Swiss cheese,
full of holes. The reason for that is when those proteins misfold, they literally cause the cellular structure of the brain to fall apart. And as you might imagine, putting holes in the sheep's brain doesn't really do a lot for its neurological function. You may have heard of a different form, bovine spongiform encephalopathy. What'd you say?
Is it mad cow?
Who said that? Good job. Mad cow disease, yeah. They think that scrapie got spread to cattle because you know that to fatten up cattle, they feed them a high protein diet, right? Well, believe it or not, once you slaughter an animal, you're left with a lot of leftovers, aren't you? Bones, guts, nervous tissue from the central nervous system. They essentially took the remnants of the slaughtered sheep, ground them up into protein powder, and put it into cattle feed as a protein supplement.
Is that why cows should really just eat grass?
Well, in the natural order of things, yes. But congratulations, we have created carnivorous cattle. Not only that, I think they actually had for a while there cannibalistic cattle. And before you all get too squirmy and start looking at things that are gross, has anybody here ever eaten Jell-O? It's just protein, isn't it? Gelatin. You know where they get it?
Beef fat.
Huh?
Beef fat.
Beef fat, beef bones. You look at a bone, you think it's just hard, right? It's calcium phosphate mostly. But it actually has a lot of protein in there that holds it together. And if you dissolve the bone in acid, you hydrolyze the protein, and it actually kind of floats to the top. And they come in with this big sweeper and sweep off that protein, freeze dry it, grind it up into powder, and there's your Jell-O.
Is that the bone marrow then?
Huh? Somebody put up a hand.
Oh, is it, so would that just be the bone marrow then?
No, it's just protein. Collagen. You've heard of collagen?
Mm-hmm.
It's collagen mostly.
Okay.
Protein's protein. And who was it that said something about cattle being herbivores? Believe it or not, herbivores will often actually eat meat if, number one, they're having some kind of dietary deficiency, and number two, they're actually able to catch it. They're opportunistic. They're not obligate. They don't have to have it. It's not normal behavior, but y'all ever heard of seeing dogs that eat dirt? Similar to that. There was a huge concern in the late 90s about whether or not the human form of this was caused by the bovine form. The human form is called Creutzfeldt-Jakob syndrome, or CJD, Creutzfeldt-Jakob disease. And no, I don't expect you to know how to spell Creutzfeldt and Jakob. What happens, the people that suffer from this have their brains fall apart. They quickly descend into dementia, behavioral issues, neurological function and motion issues. You get the idea. It's a pretty rough way to go. It's guaranteed 100% fatal. There is no treatment, there is no cure. They haven't really been able to find a definitive link between the two. The reason they think there is one is a disease called Kuru, K-U-R-U. Kuru is sometimes called the laughing sickness because people who suffered from this would laugh at inappropriate times, under inappropriate circumstances. They would exhibit jerky movements. They might stand and move in circles. They had coordination issues. The interesting thing about Kuru was it was found only in assorted tribes that were native to the island of New Guinea. And after observing this disease from about the 1940s up to the mid-60s, they realized that it was probably transmitted via headhunting. One tribe would headhunt another tribe, and they would ritually consume the corpse to take their enemy's strength and power. The government of New Guinea outlawed this practice, and the last person to suffer with Kuru died in 2006. There have been no cases reported since then. So keep in mind, we can't really truly draw a line here, can we? As to what's living and what isn't. Prion comes from proteinaceous infectious particle. They're not even calling it alive, but it acts like it is alive, doesn't it? So any questions about all that? Let's look back to the differences between prokaryotic and eukaryotic cells then. Prokaryotic cells, number one, they are generally much smaller than eukaryotic. Somewhere on the order of about 10% of the size. Now that's not to say there aren't exceptions, there are, but generally they're much, much smaller. Number two, they always have a cell wall. Always. There are no exceptions to that rule. And the third, which is the most important one, prokaryotes lack, meaning they don't have, membrane-bound organelles. Membrane-bound organelles. If you don't know what organelles are, that's okay. We will be covering that later on, okay? Before you ask, do I need to know this? Yes, absolutely you need to know this. There will be a test question that says, list and describe the three differences between prokaryotes and eukaryotes. Typically, we consider these to be much simpler cells. They're probably the ones that came first. Archies, I've already mentioned. You find them only typically in extremes. But I want you to understand that bacteria are so common because they're so tough. Does anybody know the story of Apollo 12 and Surveyor 3? You may have seen the movie Apollo 13, right? So you know those were the manned missions to the moon. Apollo 11 was the first. Apollo 12 was the second. And one of the things that Apollo 12 did was land next to a robotic probe called Surveyor 3 that had been launched 2.5 years earlier. So Surveyor 3 has been sitting on the moon for 30 months at this point. Anybody know what the temperature's like on the moon? It has a 14-day daytime and a 14-day nighttime. During the day, the temperatures get up to about 200 degrees Fahrenheit. During the night, they can drop as low as 200 degrees below 0 Fahrenheit. And y'all are thinking, we got a storm coming. So it's been doing that every month
those temperature shifts for 30 minutes. There's obviously no atmosphere. There is no magnetic field to shield it from ultraviolet light or cosmic radiation. There is no oxygen. And obviously, there's no food. Don't think anything can survive in that environment, do you? Well, they cut off the arm on the robotic probe that had the TV camera. They brought it back with them so that they could put it in the lab and see what the effects of 2.5 years on the moon would do. They cut the camera apart, they took it, stripped it completely, and one of the experiments they did was they swabbed it for bacteria. They didn't expect anything to grow, obviously. They took the insulation from the TV camera, swabbed that, put it in the incubator, and guess what happened? Streptococcus. Y'all ever heard of that? Strep throat? It had apparently survived the sterilization process on Earth, the launch to the moon, the landing on the moon, 2.5 years on the moon, the return process, and being put into a lab. Bacteria can be tough. Make sure you understand that and get that down, because this is one of the reasons why they are such concern in this infectious diseases. So what is the significance of bacteria to humans? Number one, by far and away, disease, infectious disease. Number two, industry, especially the food industry. Probiotics, cheese manufacturing, yogurt. But that is definitely a distant second to the fact that they cause disease. You could also put down research, because they are widely used in research as well as industry. So any questions about prokaryotes? All right, eukaryotes. Eukaryotes typically have 4 main groups. Everybody knows plants and animals, right? Fungi used to be considered part of the plant kingdom, but they have now been split off into their own branch, and they're generally recognized as a completely separate kingdom now. And then the fourth one is the kingdom Protista, 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 the protists. I call that the Jim Keep of classification. We like to classify things, don't we, as humans? We like to put them into categories. The problem with the protists is they don't fit neatly into any one category. So the protists can be everything from single-celled to colonial to multi-celled. Some of them are autotrophs, that means they feed themselves, they're photosynthetic. Some of them are heterotrophs, that means they have to feed off other things. Some of them move, some of them don't. So what do you do when you've got a single-celled organism that's photosynthetic and moves around in a drop of water? Plant or animal? got traits of both, doesn't it? We just stick it in the post. They have a very diverse impact to humanity. Some diseases, malaria is a good example, amoebic dysentery. Malaria alone is one of the worst diseases in terms of impact. Beyond that, you'll find some use in both the food industry and research. Food and research, and I hope you're getting that down. Produce are so variable, there's just really too many different relations to humanity to put down. Plants. Why are plants important to people? Plants are multicellular. They do not move, and they are all autotrophs, meaning they are all photosynthetic. They also all have a cell wall. All plant cells have a cell wall. By far and away, the most important significance of plants to humans is food. If you looked at the top 20 food sources that humans use, 10 of them not only come from plants, they come from one plant family, the grass family. Corn, wheat, barley, oats, I'm sure you get the idea, rice. Basically every place on the planet where civilization started up, you can trace there is a major grass-derived crop. In Central America, it was corn. In Asia, it was rice. In the Middle East, it was wheat. Fungi. Fungi are single or multicelled. They always have a cell wall. And most of them are what we call saprophytes. Saprophytes. S-A-P-R-O-P-H-Y-T-E. In other words, they're decomposers. They feed on dead things. You've got a few that are parasites in there, obviously, but by and large, they break down dead things. And they're one of the very, very few organisms that can actually break down wood. There's a substance in wood called lignin, L-I-G-N-I-N. The only enzyme that can break that down is lignase, and all fungi produce lignase. Well, most of them. In terms of direct benefits to humans, I'll turn to our mycologist.
What, for fungi?
Yeah, what do you think is the most economically significant impact?
Penicillin?
Nope, it's not medicine. Baking and brewing.
Oh, yeast.
I'm cheating, yeah, because most people don't think of yeast as being fungi, but they are. Baking and brewing.
Well, isn't the bittersweet yeast and a lot of other fungi say yeast is single celled? Right?
Yeast are single celled, yeah. But they are definitely fungi, Ascomyces. Those two activities have supplied most of human calories throughout history, period. Y'all know who built the Great Pyramids of Egypt, don't you? believe it or not, out of work farmers. In Egypt, you essentially had 3 seasons. You had the flood season where the Nile River flooded over its banks and you couldn't work the fields because it's flooded, right? Then you had the planting season after the flood receded, where you're out in the fields planting your stuff. Then you had the harvest season where you harvested all your crops. But for the 4 months of the flood season, you get to do nothing. So Pharaoh said, you know what, instead of having these people sitting around all day long, let's organize them into work gangs and have them pile up a big mountain of stone. And they actually paid them in bread and beer. Sounds like a bad plot for a bad movie, doesn't it? But it's true. Animals. Animals are multicellular, heterotrophic, meaning they always have to eat other things. They're mobile. They are capable of movement. And the last thing is they lack a cell wall. No animal has a cell wall. And those are the main cell types you will encounter. Those are the main cell types you will encounter. Is everybody cool with that? We got it? Next up, we're going to talk about the makeup of the cell. The makeup of the cell. We've already talked about the molecules, the macromolecules. Now, if you remember, I said the next step above that were something called organelles. Organelles. Organelles are specific structures in the cell with a specific function. They are structures in the cell with a specific defined function. Every cell has some organelles. Not all of them have every single one, so they can vary. The main ones you'll find in a prokaryotic cell will be the cell wall, the plasma membrane, and ribosomes. The cell wall, the plasma membrane, and ribosomes. Ribosomes are very small. They're tiny little structures floating around in the cytoplasm. The cytoplasm is just the liquid that's inside the cell. It's like a very light syrup, kind of gooey. Doesn't flow like water, but it's got a lot of stuff dissolved in it. The ribosomes are the location of translation. They are the site of translation. In other words, this is where proteins get made from amino acids. Then you've got the cell wall, the cell wall. The cell wall's most important function is protection. Protection from desiccation, which is drying out. Protection from chemicals in the environment.
protection from digestion, believe it or not. A lot of species of bacteria can form a structure called an endospore. An endospore. An endospore is just a very, very thick cell wall. Most of the antibiotics that we use against bacteria actually target the formation of the cell wall or target the cell wall itself. As a protective coating, it does pretty darn good. That's what enabled that strep to survive on the moon. Just inside the cell wall, you have the plasma membrane. It may also be called the cell membrane, but the plasma membrane is the preferred term. A lot of you have noticed I am very picky about keeping the door shut while we're lecturing, right? Why do I do that?
Stop noise from coming in.
Stop noise from coming in, you got it. If we didn't have that wall and that door, it would be a lot more difficult to teach, wouldn't it? It'd be kind of distracting. It'd be difficult for you to learn. So having that wall with the door in it allows me to control access. That is essentially what the plasma membrane does. It controls access. I want my students to stay in the room while class is going on, right? I want people out there to stay out and not disturb us while class is going on. But at the end of class, we all want to go home, so we open the door and we're able to leave. Notice that the door allows controlled access. We could lock it to keep people out, right? So everybody got that? You understand what the function of the plasma membrane is? We'll go into more detail in it in a minute, but I want you to understand that the cell wall is basically just protection. It's armor and it keeps bad things out. The plasma membrane is more selective. It allows you to keep certain things in and certain things out. And I don't see too many people taking notes. That's kind of scary. What is left is basically just the cytoplasm. The cytoplasm. The cytoplasm, like I said, is the solution that is found inside the cell.
So I guess I can sit back and enjoy it at a time, right?
You got dissolved sugars in there, proteins, ions, DNA, and it's all just kind of floating around in this big soup. You also have another structure called the cytoskeleton, C-Y-T-O, skeleton. Cyto is a word or a prefix that means cell. So this is the skeleton of the cell. Now it's not made up of bones, obviously. It is just a network of tubes and fibers, a network of tubes and fibers. It helps give the cell shape, it also acts as an anchor point. You can attach proteins to it and other things. And finally, it assists in movement. It assists in movement. Everybody with me? Next up, we've got eukaryotic cells.
What was that?
Eukaryotic cells. Now, pretty much every thing you find in prokaryotes, you will also find in eukaryotes. Now, again, though, there are differences between some of them because, like animal cells, won't have a cell wall. They will have a cytoskeleton, cytoplasm, plasma membrane, and so forth. Everybody got that? So starting at the center, you have a structure called the nucleus. The nucleus. If I were to ask you what the function of the nucleus is, some of you who have recently had high school biology might say it's the control center of the cell. Every time I hear that, I want to slap the person who first said that, because it is not a control center. Most of the genetic regulation actually happens outside the nucleus. What it is a library, an information depository. The only thing it does is store and protect DNA. That's all. It stores and protects DNA. Why is that important? Well, let's say that this was not a biology class, but it was a woodworking class. And I give you an assignment over the weekend to build a birdhouse. And I'm assuming every one of you have a fully equipped wood shop at home. So, but you have to build my birdhouse to my blueprint. So I put the blueprint over on reserve in the Learning Commons. But I only have one copy of it. If you want to make a copy and take it home, you have to go over there, sign in, get access to it, and either photocopy it or take a picture with your phone, put it back up, and leave. You see how, if that's only one copy, you want to protect it, don't you? And that is what the nucleus does. It stores and protects that blueprint, prevents damage to it, controls access to it. Extending away from the nucleus, you have a network made up of membranes. Anybody ever been in a hospital where they got those curtains and they can pull them and isolate part of the room? It's not a total isolation. It just prevents people from seeing what's going on. And you can reconfigure it, can't you? This network of membranes is kind of like those curtains. It partitions off a segment of the cytoplasm for something. This network is called the endoplasmic reticulum. The endoplasmic reticulum. Will everybody be happy if we just refer to it as ER? Whenever I talk about the ER, I'm talking about the endoplasmic reticulum. So we're all okay with that? And yes, you can answer it that way on the test. Every biology course I've ever been in or taught, it always ends up getting abbreviated ER. So everybody cool. It comes in two flavors: rough and smooth. Rough and smooth. The only difference between those two, the rough ER is studded with ribosomes. Those membranes have ribosomes bound directly to them. The function of the rough ER, as you might have guessed from the presence of ribosomes, is protein synthesis and processing. Protein synthesis, processing, and assembly. So you've got messenger RNA coming in, you've got transfer RNA coming in, bearing amino acids. They're binding to the ribosomes, it's making this peptide chain. Other enzymes are chopping that peptide chain up, folding it over, and assembling it. And essentially, you've just curtained off an area of the cytoplasm where you can do all that work more or less uninterrupted. Does that make sense? The smooth ER is basically the same thing, but instead of proteins, it's lipids.
It's what?
Lipids. Fats, oils, waxes. Now notice here, all of this is a membrane, right? That membrane is what helps partition off the cytoplasm. The nucleus itself has its own membrane. It's called the nuclear membrane, so you may want to get that in. You won't find either of these structures in a prokaryotic cell. You won't find either one. So these membrane-bound organelles are characteristic of a eukaryotic cell. The next organelle is called the Golgi body, the Golgi apparatus, or just the Golgi. For a very long time, we really didn't know what this thing was for. But in the last 40 years, we've more or less figured it out. And for lack of a better term, I would say it's kind of like an Amazon fulfillment center. If you order something from Amazon, somebody pulls it off the shelf, don't they? Then they package it up. To do that, they have to get the appropriate size container, put any padding in it that it might need. Then they address it, don't they? If you don't have the address, you can't send it to the right place where it goes. Then, and only then, do they throw it on the truck. So basically, the Golgi is doing the same thing with macromolecules. If you've got a cell that makes a hormone, it's got to get that hormone out of the cell into the bloodstream, right? So it's going to take it out of the smooth ER, and it's going to wrap it up in part of the membrane of the Golgi. Then it's actually just going to pinch off that little sealed packet. It's now got its own membrane, doesn't it? It goes into the plasma membrane where it fuses and releases it out into the bloodstream. And that is the function of the Golgi: packing and shipping. And like the ER and the nucleus, it obviously has its own membrane. Next up, we have lysosomes. 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 Lysosomes contain digestive enzymes. Why is that important? Well, some cells actually take other things in and break them down, right? Your white blood cells, they engulf viruses, they engulf bacteria. Once they do, they want to break those invaders down, don't they? So instead of releasing digestive enzymes within the cell, they essentially have just a little fluid-filled balloon. Yeah, it's got its own membrane. And they stick the food particle inside that, or they stick the invader inside that. The reason you want it walled off like that, you don't want those digestive enzymes free in the cytoplasm chewing on your proteins, do you? They actually can rupture and self-digest a cell. That actually happens more often than you might realize because it recycles the molecules from a dead or dying cell. Does everybody see how that works? Then you've got something that's very closely related to it called a peroxisome. Peroxisome. Just like the lysosome contains digestive enzymes, the peroxisome contains enzymes that break down toxins. So if you get some kind of toxin introduced into the cell, you don't want it bouncing around destroying things, do you? So you put it into the peroxisome, which number one, walls it off. It's now safe. But you don't want it getting out, so you break it down inside there. Next up, mitochondria. Mitochondria. Again, in high school, they probably call this the powerhouse of the cell. That is a terrible function for it. Its function, its only function, is to make ATP. ATP stands for adenosine triphosphate. Tri means how many? 3. ATP is kind of like cash. I know very few of you probably use cash today, am I right? Most of you tap your phone or something like that. I'm always afraid to do that because of my lack of knowledge, I'd probably end up hooking my bank account to every electronic device in a 5-mile radius. But back in the old days, we didn't have all that. We had cash, right? If I wanted to stop by and put gasoline in my vehicle on the way home, could I use cash to pay for it? If I wanted to run by Wendy's and get myself a Baconator combo, could I use cash for it? Hypothetically, I could stop by the water department in City Hall on my way home and I could pay my water bill with it, couldn't I? You see where I'm going with this? Cash can be used to pay for anything, for any purpose. I have somebody come mow my lawn, I pay him in cash. See where I'm going with it? ATP is like that. It is used by the cell for anything that requires energy. Want to make a protein? Boom, ATP. Want to break down a damaged protein? ATP. Want to move something outside the cell? ATP. Want to duplicate the DNA? ATP. I think you can see why ATP production is the most important function. So everybody got that? The mitochondria also have their own membrane. And interestingly enough, they also have their own DNA. And that DNA is actually different of what you find in the nucleus. Maternal DNA inheritance. It's only inherited from the mother. You want to get that down. Mitochondrial DNA is always inherited from the mother. I was like, why? Good question. It's because who supplies the cytoplasm and organelles of the very first cell in an embryo? Mom or dad? Mom, because the egg has everything it needs except a second set of chromosomes. Sperm are basically nothing more than a genetic information packet with a motor attached. I'm serious. As my old cell bio prof used to say, sperm are cheap, eggs are expensive. But what it amounts to is the mitochondria in my body came from an egg that my mother had, right? So essentially, my mitochondrial DNA will be identical to her mitochondrial DNA. And her mother. And her mother. And so far. And they can trace it back pretty good ways. It's actually been used to help solve a lot of crimes. There's one particular discovery, it was announced about 7 or 8 years ago, and it keeps making the rounds and popping up in the headlines once in a while. Supposedly, and I do say supposedly because I wouldn't put my hat on this, I think they actually solved Jack the Ripper through mitochondrial DNA. Supposedly. The story is that this particular piece of evidence was a shawl, you know what a shawl is, that one of the victims was wearing that a cop had picked up at the murder scene and kept in his family, and they passed it on down through the years. And they finally tested it for mitochondrial DNA, and they tested a maternal descendant of the guy that they thought was Jack the Ripper, and he came up with a match. Yeah, I frown too, because there's this little thing called chain of custody. There's no way to prove that that particular shawl was at that particular crime scene, right? For all I know, they went out and bought a dish towel at a local Walmart, dirtied it up, and said, here you go. So DNA doesn't lie, but it also can't tell you where it came from if you don't know where the substance on there came from. Everybody got that? We'll talk a little bit more about mitochondria in a minute, but I want to move on. Next up, we've got the plasma membrane. Now, the plasma membrane of a eukaryotic cell is similar to what you see in prokaryotes. But keep in mind, you may or may not have a cell wall outside it. It is in the form of a phospholipid bilayer. Phospholipid bilayer.
How do you spell that?
P-H-O-S-P-H-O-L-I-P-I-D. Phospholipid. Phospho means phosphate. That's what these little circles represent, phosphate groups. This is the outside of the cell. This is the inside of the cell. Everybody see that? These two little things coming down are a pair of fatty acids, 2 fatty acids. Sounds like a lipid, doesn't it? Now you see why we call it a phospholipid. I don't see anybody writing that down. The phospholipid bilayer gives it some very unique characteristics. Phospholipids are amphipathic molecules. Amphi is A-M-P-H-I, pathic, P-A-T-H-I-C. An amphipathic molecule is similar to a polar molecule in that you've got different properties on different ends. The phosphate group is what we call hydrophilic, P-H-I-L-I-C, hydrophilic, water-loving. It interacts very, very, very well with water. Lipids, as you know, are hydrophobic, waterproofing. And basically, this allows the cell to control what goes in and out. Transport across the membrane. Okay, there are three different forms of this. The first is called diffusion, D-I-F-F-U-S-I-O-N, diffusion. Let's say I had a container of some substance that is really, really, really smelly. Good or bad, it's just strongly scented, right? If I take the lid off of that, where is the highest concentration of it going to be? Here or back there? Bingo. Who will likely smell it first? Probably me, then Carol. Because it's diffusing out. It's going from an area of high concentration to an area of low concentration. High to low. Eventually, let's say we kept the windows shut, we don't have the air circulating, we come back at the end of our lab period at 5:30, the whole room is going to smell equally, right? Because it will have reached a state of equilibrium. This is what we call a concentration gradient, a concentration gradient. You go from an area of high to low. It is passive transport. That's your first type. Passive transport. Passive because it does not require energy. When I get home from work tomorrow, I'm going to have to be sure that I park my vehicle at the bottom of my driveway because I live on a hill. We're supposed to get what? A lot of snow, maybe ice, right? If I park up at the top and don't set the parking brake, leave it neutral, my vehicle's going to want to slide to the bottom on it very easily. It's going from a high energy state down to a low. Does everybody see that? High to low. If I park it down at the base, right there at the little pull in by the mailbox, chances are pretty good it's not going to slide anywhere because it's level. Does everybody see that? We're not having to fight against the slope. The second form is what we call active transport. Active transport. Let's say that instead of parking at the bottom of the hill, I'm stupid and I park it at the top and it slides down, right? And I want to get it back up there. So what I'm gonna have to do then is get all of my students and pay y'all pretty well to push it up the hill, right? That can be a hard job. It's gonna cause you to expend a lot of energy with it. See how we're going against the gradient? Same principle, that's active transport. If we are doing active transport, it is going to cost us energy. Active transport is going to cost us energy. Well, that's pretty straightforward, isn't it? There's both active and passive. What is in the middle? Well, this is an interesting thing. It's called semi-active transport. Let's say that my garage is at the bottom of the hill. It'd be pretty easy to get my car into the garage, just roll it off, right? You'd roll right down in. Not with the garage door down. I'd slam into it and I'd have a messed up bumper and a dented garage door and a very, very hacked off motor. So what would I need to do? I would have to open the garage door. Does that cost energy? Yes, it does. Have to do that before I could just let it roll in. Does everybody see where I'm going with this? There are certain molecules that will be higher concentration outside the cell, but they can't get in because they're too big, because they're nonpolar, because they have a different charge. So let's say that I'm wanting to get my marker from here to the desk. Easiest way is just to let it drop, right? I open my hands and it falls. But if I've got a barrier down here, I have to change the position of the barrier, don't I? Does that cost me energy to open my hand like that? Not a lot, but it does. That is semi-active transport. It's the same direction as diffusion, high to low. But it requires you to spend some energy to open the gate. Everybody got that? And that is semi-active transport. The technical name for it is facilitated diffusion. Facilitated diffusion. And who has the sign-in sheet? Did everybody sign? Okay, thanks. Think that's a good place to stop? Y'all don't? I can talk for 2 more minutes. Is that a good place to stop? Nobody will complain if we get out 2 minutes early. I want to make sure you get your money's worth. I'd hate for you to not get those 2 minutes you paid for. Stay safe, stay warm. See you either in lab or next Tuesday.
If it snows and like we're on class